Among the recorded plagues in human history, the Spanish flu, which was a pandemic between 1918 and 1919, isolated more bacteria than humans who died in the Spanish flu. Researchers at the time believed that one of them was the causative agent. A vaccine against PAFL was developed in Japan and was inoculated to more than 200,000 people between 1919 and 1920. In other words, at that time, the existence of the virus was unknown, and a vaccine against misguided bacteria was being developed based on a completely wrong thinking pattern. It being to be wrong about the reason for something or the way to achieve producing the vaccine, I think it's barking the wrong tree. Very mysteriously, the vaccine was less effective in stopping the disease but more effective in reducing mortality. Probably, it prevented the secondary infection of bacteria invasion of influenza. The cause of the Spanish flu was unknown after all, but it seems that it disappeared spontaneously in about 40 years and ended. Influenza is a local infection in which the virus infects the mucosal epithelium of the respiratory tract, which directly leads to its onset. Originally, the effectiveness of vaccines against local infectious diseases is low, and influenza vaccines are one of them. In the development of a seasonal influenza vaccine that is prevalent every year, generally speaking, it takes more than half a year from the preparation of chicken chicks to adult chickens. And it takes more than half a year to commercialize the fertilized eggs produced by this vaccine. It takes half a year required for fertilized chicken eggs for the production of influenza vaccine. Currently, there is a method of concentrating the influenza virus propagated in a growing chicken egg with a high-speed centrifuge. Unlike eggs as a common food item, poultry farmers must set up special poultry houses to raise hens and roosters together for influenza vaccine production. The number one factor in the annual influenza epidemic is the frequent mutation of influenza virus antigenicity. Due to the large number of variants of influenza virus, the virus strains targeted by the seasonal influenza vaccine, which is prevalent mainly in winter, are changed every year. Influenza virus is the cause of influenza, but it is mutated every year and divided into many subtypes. Vaccines with antigenicity that are predicted to be prevalent in the next influenza season will be given, but high preventive effects cannot be expected in years when the prediction is not met. At present, seasonal influenza is transmitted to all age groups worldwide and is in a state of repeated epidemics all over the world. Influenza virus infects the mucous membrane of the respiratory tract, so immunity, mainly antibodies, cannot be prevented. When the vaccine is injected, antibodies that block the infection of the virus appear in the blood, but only part of it seeps out onto the airway mucosa, about 1/100th of the amount in the blood is there. Therefore, in order to directly immunize the respiratory tract, a method of vaccination by nasal or inhalation has been tried for some time. The effect of an activated influenza vaccine on preventing the onset of influenza is 25 to 60% in children and 50 to 60% in adults. It is believed that the influenza virus has been in waterfowl for millions of years, resulting in a coexistence relationship between the two. That is, it is assimilated. Waterfowl are asymptomatic with no pathological symptoms when infected. There are researchers who explain the migratory bird theory about the source of influenza as follows. Influenza spreads by airborne infection, so it can be said that it is a virus that spreads widely in densely populated cities. Influenza viruses are thought to be lurking in frozen lakes and swamps near the Arctic Circle, such as Siberia, Alaska, and Canada. When waterfowl such as migratory ducks and cancer return for breeding in the spring, the virus sneaks into the waterfowl's body and propagates in the intestinal tract. What is the path by which the influenza virus in waterfowl droppings can reach humans? Pigs have an important mediation for that. The cells of the respiratory tract of pigs are capable of infecting many viruses, and when the waterfowl comes here, a strain of mutant that infects humans is born. Pigs can be said to be the manufacturing factories for influenza viruses. According to this theory, migratory birds do not develop because they can coexist with the virus and disperse feces on their way between breeding and wintering areas. When the virus is taken up by mammal pigs, it becomes easy to infect humans, and influenza spreads in winter. In order to infect, the virus first attaches to sugar-containing receptors on the respiratory surface of animals, but human influenza virus receptors have a different structure from that of birds. So they are very difficult to attach to. However, pigs have not only avian influenza virus receptors but also human influenza virus receptors. According to this theory, people are the first source of infection in Southeast Asian countries, such as those living with waterfowl and pigs. But pigs cannot be left unchecked in urban areas in developed countries. Pig farms are usually located far from urban areas. With the new coronavirus infection, the borders of people entering and exiting foreign countries are almost abandoned, and is it easy to infect humans from pigs and spread? Therefore, the source and transmission route of influenza virus are often unknown. However, in the case of influenza virus, it becomes high fever after infection and is accompanied by inflammatory symptoms of bronchi and lungs, and since there are therapeutic drugs, it can be completely cured by medical treatment for about a week, and the virus is affected by the action of the human immune system. It is thought to disappear naturally from the body. There is also a report from the Japanese Society of Clinical Physicians that the effectiveness of influenza vaccines is decreasing year by year. According to this report, the effect was significant in all age groups from the 2001-2002 to 2007-2008 seasons, but not significantly thereafter except in some 2009 and the 2014-2015 seasons. Is said to be, if we can develop a universal influenza vaccine that is effective against all influenza strains, we will be able to stabilize the supply of vaccines and prevent pandemics of influenza caused by viral mutations. Therefore, it has been the subject of research for decades. In order to realize this universal influenza vaccine, the idea of creating a vaccine by targeting another antigenic site, host-doc, where antigenic variation does not occur, instead of the viral antigen, HA-head, targeted by the current vaccine. There is a genetically modified vaccine that links 9 specific regions that do not mutate from 3 types of influenza virus proteins has been announced as the universal influenza vaccine. After that, in 2015, the results of a mouse experiment of a vaccine prepared by another method were announced. without neutralization of antibody. Microorganisms such as bacteria and viruses, and toxins such as snakes and insects bind a part of their structure to the cell surface and invade the cell to show toxicity. If the antibody binds to the part that binds when invading the cell, the microorganism or toxin cannot bind to the cell and cannot show toxicity. In this way, the antibody may reduce the infectivity of microorganisms or reduce the toxicity of toxins by binding. For example, influenza virus binds hemagglutinin on the surface of the virus to sialic acid residues in airway epithelial cells and invades the cells. So antibodies against hemagglutinin reduce the infectivity of influenza. This is called a neutralizing action. In 1976, Tonegawa et al. Discovered the phenomenon of gene rearrangement of immunoglobulins 17-18, and answered the genetic level mystery of the diversity of this antibody. Other phenomena such as somatic hypermutation, gene conversion, and class switch recombination are also known to be involved in antibody diversity. Development of avian influenza vaccine using reverse transcription technology of genes. Immunoglobulin antibody. The light-colored part is the light chain, and the black part at the tip is the variable part. The matching antigen specifically binds to the variable region. For example, ChAdOx1nCoV-19, the new coronavirus vaccine developed by Oxford University, uses adenovirus. The mechanism by which ChAdOx1nCoV-19 vaccine works is to collect the gene corresponding to the protein of the spike on the surface of the new coronavirus. This gene is integrated into the gene for a common cold virus, adenovirus, collected from chimpanzees. Adenovirus is detoxified so that it does not multiply in the human body. This is the vaccine. In other words, it seems that the adenovirus incorporates a protein fragment of the receptor domain portion of the spike protein. It is wondering why adenovirus can be used as a vector to invade human cells to form surface antigens. Moreover, the mechanism by which it can be expressed as an antigen is unknown, and it is considered that there is no reason for it to have antigen activity. However, when this vaccine is injected into humans, immune antibodies such as antibodies and T cells are produced. Neutralizing antibodies are antibodies that recognize the binding site of viral proteins to human cell receptors in particular. Therefore, in many cases, the neutralizing ability can be exerted by blocking the receptor binding of the virus. Now, I can say strongly that human beings may have developed a vaccine against a misguided virus under a completely wrong thinking pattern. Maybe many researchers are acting to be wrong about the reason for something or the way to achieve to producing effective vaccine. I think it's barking the wrong tree. This is because they are vaccinated completely ignorantly and unknowingly about the intracellular and extracellular expression of proteins for antigen recognition and the maintenance mechanism of the complex three-dimensional structure by glycoxylation. It is said that when a new coronavirus is encountered, antibodies and killer T cells are convened to attach to the virus and repel it, but it is unknown preventive effect. It sucks.